become a problem, and how you separate those out, I think, is our responsibility as the adult society, trying to provide alternatives and restrict situations that induce harm. That would lead to my presentation. The endogenous cannabinoid system, I would like that to be a common parlance. I would like every person in this community to understand this. And in that regard, I was approached by a fellow yesterday. There was an international cannabinoid CB2 conference in Banff. And there's a chance that we can bring the second annual to Mendocino. And here you have a group of people from all over the world coming together for 3 days to do nothing but talk about one of the cannabinoid receptors. The CB2 receptor is found in the T cells, the B cells, the lymph nodes, spleen, thymus, and one of its primary functions is in modulating the immune system. And if you know anyone that uses cannabis and benefits from it, there's probably a 60–70% chance that they have an inflammatory or an autoimmune disorder. They've got rheumatoid, they've got osteo, they've got post-traumatic arthritis that turn into an arthritis. And the 5 different anti-inflammatory compounds in cannabis, which don't go on to block the enzyme that protects the stomach, that leads to the GI bleeding of the ibuprofen and more commonly used problems for arthritis. Cannabis is wonderful because there's been 34 million years of research put into the development of this particular plant, and it interfaces very well with the physiology in the human being. And it goes back to this picture here. This is the receptor, what they call the serpentine receptor. It's embedded in the membrane of every cell, and I say every cell with some degree of anticipation. At this point, we have CB1 in the brain, CB2 in the immune system. A new receptor called GP55 is going to be the 3rd cannabinoid receptor. GP12 is going to be the 12th or the 4th receptor. And it looks like there's likely to be a protein receptor in the membranes of a tissue type that's trying to coordinate and regulate its activity. But going back historically so we can get a sense of the origins of this system, there's an organism called archaea, which is a very primitive single-cell organism. We had to create a whole kingdom because this organism is so unique. It's not a plant, it's not an animal, it's a very, very, very simple organism that's billions of years old. But in the membrane of archaea is this protein that is in the cells of our immune system. It's in the cells of our brain, it's in the cells of our intestinal system. It regulates glucose metabolism in our body. This is an epiphany of conservation. It was such an effective design that 4 billion years later, this exact same serpentine protein is involved in the regulation of cellular function. This here, I don't know if you can read it. It lists the cell types where the CB2 receptor is found. It's that structure that we just looked at previously. We know every amino acid that goes into this protein, and there's a slide coming up that was a computer modeling of the function of this particular receptor and how it, in fact, the active ingredients move through the lipid medium of the cell membrane and attach inside the cell membrane to this receptor, and it can direct the cell to go through an apoptotic response, which is where it can tell the cell to lyse the membrane around the nucleus and destroy the cell. And this is useful because 1 million times a day a cell goes precancerous. Body is highly competent at dealing with that change, and what the cannabinoids do is they identify that cell. CB2 receptors in some situations are not present in some normal tissue types, but when the cell becomes abnormal, this receptor is produced, installed in the membrane, and brings the attention of the immune system, which can range from additional assistance to destruction if it's deemed that that cell is a precancerous cell. So all this bears to why it would be so exciting to bring this conference to Mendocino, because we are in a position to help undo some of the damage that's been done to this plant and these 66 different cannabinoids that are found in this plant that are unique in that, I mean, what are the chances that you're going to produce a large molecule that binds to the receptors in our body and can be involved in regulation of glucose metabolism, pain reception, seizure potentials? I mean, it's absolutely huge. And if if you have some extra time, you can go to ICRS, ICRS, go to the 2008 Symposium page, and download the abstracts. It's a 30 meg PDF, so be on a high-speed line. It's about 1.25 inch book. There's 140 or 50 original research papers in there. This one came out in June of '08. The one in '07 is equally impressive. And what you'll find in there is that the rest of the world is amazed at how the body's endogenous cannabinoid system interfaces with these plant cannabinoids. And what the bottom line is, the body produces these endogenous cannabinoids to help modulate function. They're a feedback molecule which can either increase or decrease activity, but with the intent of restoring optimal function. And it's an excellent design that here's a plant that supplements our endogenous cannabinoids, and we've produced 5 or 6 of these chemicals that bind to this receptor that help restore normal function. And what they do is they come in and facilitate that optimal regulation. Further, there's a class of compounds called terpenes, which they bind to that CB2 receptor and change how tightly it attaches to the cannabinoid. And when I say cannabinoid, I should probably call it an icosanoid. That way we can remove that whole word